Okay, folks, last video for DNA replication. Hopefully we're going to bring it all together here. Remember that we've got our replication bubbles, our forks created by helicase and topoisomerase, and then we got our primase comes in, DNA pol 3, DNA pol 1, you got your ligase. I mean, you got all kinds of enzymes there working together. We're going to bring it all together and kind of back out and see kind of a bigger picture here now, okay? So generally speaking, if you have DNA that's just in a long strand, what really happens here, because the DNA is way too long, there's not just going to be one replication bubble and it works in both directions. To give you an idea, helicase works at 900 base pairs per minute. So it unzips this thing 900 base pairs per minute. Well, some of these chromosomes are millions of base pairs long. It would take forever to duplicate all of the DNA. It would take forever. So the strategy, the approach, the solution is there are multiple replication bubbles that always form on a strand of DNA. So I kind of crappily drew a strand of DNA here, but I try to illustrate that there's multiple replication bubbles. And hopefully you're recognizing that I've drawn in here or hinted in, this is the leading strand, and then there's going to be a bunch of lagging, Okazaki fragments here on the lagging strand. And ultimately this thing is just building, and this one's just building, and this one is just building. And eventually the replication fork here will meet the replication fork here. All the enzymes will fall off and both sides are duplicated. So you end up with a duplicated side over here. So notice where I'm pointing out here. You end up with a duplicated side on the top and a duplicated side on the bottom. And eventually you're going to end up with then, ultimately, when all the bubbles meet each other, you're going to end up with a side on the top and a side on the bottom. And if you hold it together in the middle with a little band of protein around the middle, and hopefully you're like, aha, you kind of recognize this, this would be your centromere, right? That would be your centromere right there. And the overall thing would be a duplicated chromosome, and we'd just draw it out as an X. So we'd show that little centromere right there, and we would just draw it out as an X, and then each duplicated side is now called a chromatid. We're not drawing out all the detail down here. We're not drawing out the fact that this little, sketch right here includes a double helix of part old and part new DNA. And we're not showing that this little sketch here of a single little noodle is actually a double helix with part old and part new. And so ultimately, that's the big picture that you need to see. So when mitosis occurs, when meiosis occurs, the first step is always, replicate your chromosomes, duplicate your chromosomes, look like an X. This process goes on somewhere around 300 million times per hour inside of your body. Unbelievable machinery at work here just to duplicate your chromosomes, okay? Just to pass down that information. There is some flaws. There's one major flaw that dooms all life on this planet. And I like talking about it because I find it pretty interesting, and I think it kind of ties in. I don't think it does tie in with what we're talking about right here. So hopefully you understand. This is a picture from one of our textbooks. This is the leading strand up here. And this is the end of the chromosome. So like this is the template strand, and then this is the end of the chromosome where polymerase 3 would just zip along, and it would eventually just fall off on the tip because it got the primer somewhere up here, and then it just started and it went in this direction and it just kept going and going and going. On the opposite side, we've got those Okazaki fragments, remember? So we've got a primer laid down every once in a while, and then we've got our DNA strip laid down. And then we had a primer here that got replaced by DNA pol 1, and then we have our strip laid down. And then we had a primer here. And remember, each time this thing had to jump back each time and lay new primers. This thing the primase had to keep going back as this thing kept opening up. And the last couple of primers that get laid down, they take this box and they're trying to show this box right here. So they're trying to show that this is the last couple of primers here, okay? So the last couple of primers. Here's the problem. DNA pol 1, read this carefully and think about it. DNA pol 1 can only work when it finds an exposed 3' end of DNA to attach and start working. So think of it. DNA pol 1's job is to find RNA, but it actually doesn't find RNA. It doesn't find the primer. The chemical cue for DNA pol 1 to say, oh, there must be a primer here, is the fact that there is a 3' end right here on DNA where the pol 3, remember it worked in this direction here. Sorry for my scribbly writing here. It worked in this direction from 5' to 3', but it just bumped into that primer, and then the pole 3 fell off, and it said, okay, I guess I'm done there. And pole 3 just goes and finds another spot to work. But DNA pole 1, remember it's spell checker, right? It identifies the 3' end and it bonds on there. So, and then it works in this direction and it replaces all of this RNA with DNA. But it needs a 3' end of DNA to start, to like give it a clue, to give it a chemical cue that like, here, this is where you hop on board, okay? So what's the problem? The last primer, the very last primer that's laid down on the tip of a chromosome doesn't have any DNA before it because it's the last primer. There's nothing over here. There is absolutely nothing over here. And so what ends up happening is that this primer never gets replaced. Well, what happens to RNA after a few minutes of just sitting on there without getting replaced? RNA doesn't bond very well with DNA. It doesn't like hold together. It doesn't stick like glue. Like it's just not very good. It's not a very strong bond. So what ends up happening to it? The primer just falls off. So the whole primer, that chunk that was this red, it would be red. I'm sorry for drawing this in black here, but this red piece right here just eventually just falls off. Well, what's the consequence? The consequence is that you have lost DNA. You've lost 5 to 10 nucleic acids off the tip of your chromosome every single time that you go through mitosis. So what's the big deal? Well, remember, chromosomes are DNA, right? And DNA has genes. Maybe there's a gene right here to here. Maybe there's a code in here somehow that makes a protein, and it's important. Well, when we divide one time, we lose 5 to 10. When we divide again, we lose another 5 to 10. The next time this happens, we keep just chopping into this thing, and eventually we get to a gene that causes a problem. So the result of this flaw is shortened DNA. And again, this happens every time the chromosome is duplicated prior to mitosis, or meiosis, really. And eventually the eroding DNA, that part that falls off, reaches a spot that's critical. It reaches a gene. When you lose a gene, sometimes it's really consequential. Sometimes it's not a big deal. But when an organism loses a gene that's important, and I'll say that is important, okay, it programs that cell to just apoptosis. It just says, just destroy yourself. Just be done. You're not working right. So it just commits suicide, basically. That cell does. So what happens when enough cells do this? Then a tissue can fail, an organ can fail, a whole system can fail. Death occurs. Ultimately, this is why you die of old age. This is it. Like, this is why you can only make it so many cell divisions in your lifetime. This is why not just you, but every living thing on this planet has the same exact stupid flaw in it. It all comes down to DNA pole 1 dooming you because it can't figure out how to replace this. If it could, your tissues wouldn't get old. That's a crazy concept. Ultimately, every living thing on this planet has a different length of DNA at the end, at the tip. That is important. Those lengths are called telomeres. They're called telomeres, okay? So those extra lengths are called telomeres at the end. And telomeres are just gibberish DNA. So it's intended to act like a buffer. Like, okay, yeah, we're going to lose DNA every time we divide. So we'll just put a whole bunch of gibberish DNA at the end. It might be 5,000 letters long. It might be 5,000 nucleic acids long. And, it's like, okay, we'll lose 5 this time. We'll lose 5 next time. We'll lose 5 next time. And then that gives you a longer lifespan. So organisms that have short lifespans have short telomeres, have short gibberish sections at the end of their DNA. It's like a mouse that maybe only lives a couple of years. It's got short telomeres on it. Humans, longer telomeres. Turtles, really long telomeres. Trees, incredibly long telomeres. They can live thousands of years. So every living thing on this planet has different length telomeres. Now, there's one place that you would want to avoid this, and there's actually a failsafe. There's actually like, okay, we'll save it here. There's an enzyme to avoid that, and that enzyme is called telomerase. And telomerase is an enzyme that works on telomeres and replaces the lost parts. So basically conserves you forever. Where would you need this? And I'll give you a second to think about it. Where would you need that? You wouldn't need it in your heart and your lungs and your, organs that you'd normally think of. Your brain, nope, that'd be great, but nope, sorry, you're going to lose DNA there. But where can species not lose? They can't afford to lose any DNA in their reproductive cells. So in your ovaries, in your testes, when you're making eggs or sperm, telomerase is an enzyme that's active in those areas that's always replacing the length of DNA. Because if you pass down a shortened length of DNA to your kid, you've already shortened their lifespan. And if they pass down a shortened DNA piece, then within like 10 generations, you don't make it long enough until sexual maturity. So that's how we have overcome. And all living things on this planet, their reproductive tissues have telomerase. There's one organism out there, and I put them on here, this is the immortal jellyfish. We've discovered a species of jellyfish that actually does this in all of its cells. We believe that these jellyfish have never died, that they just basically hit a reset button, but they are probably millions of years old individually. Pretty cool. All right. Some review questions for you. I'm not going to answer them, but these should be the things that you guys should be able to answer now after you've watched all the videos and I'll say suffered through this, but I hope that has been educational for you. So name the enzymes, explain the importance of the 5' 3', leading the lagging strands is really important. It's going to come up on your AP test over and over again. And then can you describe why people or old things die from old age? Okay. That is it. Thanks for paying attention, folks. Appreciate you.